be talking a little bit about enzymes. This is a pretty short lecture. Hooray. All right. So enzymes, as we saw in the Amoeba Sisters video, enzymes are proteins that speed up the chemical reactions of the cell. They do not cause the reactions to happen. They simply speed up reactions that will already occur. Without enzymes, the reactions of the cell would proceed so slowly that the cell would actually die. So these enzymes are very specific. They can only carry out one job, but they do that one job extremely well. And enzymes are never used up in the reaction. They can be used over and over again. That's what keeps the cell able to stay alive. Okay, so how enzymes work. An enzyme comes together with a substrate and forms the enzyme-substrate complex, and that releases the enzyme to do that process again, and then the products of that chemical reaction. So if we look at this and we say this purple thing is the enzyme, these are the two substrate molecules. So the enzyme could be putting two things together, it could be breaking something apart. It's just helping in whatever chemical reaction that is. Okay, the enzyme substrate complex is formed where they all become like one piece. And then the chemical reaction takes place and the products are released. And that's supposed to be showing you that from red and green it turned into blue and yellow. So somehow there was a chemical reaction that made them different. And note that the enzyme is unchanged and can be used again and again and again. So the substrate molecules are the reactants in the reaction. So they're like the ingredients. Like if you're making a cake, right? The reactants are the ingredients. And the products are the new substances that's formed. So that'd be like the cake. Does that make sense? Thank you. So we call this little pocket where the substrate fits in and the products are released from is called the active site, because that's the part that's actually going to do the chemical reactions. So the active site is where the substrate molecules bind to the enzyme, and the fit is so precise that is compared to a lock and key. This idea of a lock and key fit is like the actual explanation that biologists use for how specific enzymes are to their substrates. All right, so remember, the shape of the enzyme is so specific that only one shaped substrate can fit. A specific enzyme is required for each reaction in the cell, and there are over 2,000 known enzymes that work inside each and every one of your cells. So the enzymes are not used up in the reaction, and they are available to do the reaction again. And that's pretty much everything you need to know about enzymes.